Ovarian cancer is the sixth most common cancer and cause of cancer death in women in the UK, with 7,500 cases and 4,200 deaths every year. Ovarian cancer occurs when abnormal cells grow in the ovaries and form tumours. In the UK, 35% of ovarian cancer patients survive by 10 or more years, with treatment options including surgery and chemotherapy. Due to its relatively poor prognosis, research into ovarian cancer and its treatment or therapy is a very active area. One such research paper is the topic of this video, where the scientists investigated whether an already approved set of drugs called Pan-HDAC inhibitors could be used to treat ovarian cancers carrying a certain mutation in a gene called ARID1A. So what does the ARID1A gene do and why is it important? The ARID1A gene encodes a subunit of the SWI/SNF chromatin remodeling complex, which regulates transcription by altering chromatin accessibility in an ATP-dependent manner, as indicated in this figure. Genes encoding subunits of the SWI/SNF complex have an average mutation frequency of 20% across various cancer types, and for ARID1A this can be even higher. The majority of mutations in the ARID1A gene are frameshift or nonsense mutations, which lead to the loss of ARID1A protein expression. To understand why mutations in a gene involved in chromatin remodeling can contribute to cancer, we must first understand what chromatin is and its function in the reading or expression of genes. A cell's DNA containing all of its genes is packaged into the nucleus with the help of proteins called histones, and this combination of DNA and histones is known as chromatin. For genes to be expressed, they must be packaged more loosely within the chromatin so the cell can access and read them. And if a cell cannot regulate chromatin accessibility via chromatin remodeling, as is potentially the case upon ARID1A mutation, they consequently cannot correctly regulate gene expression, and this can cause many problems and may contribute to the development of cancer. Another important aspect of chromatin accessibility is the chemical modification of these histones, i.e., when certain chemical tags are added to them, it can influence how tightly packaged the chromatin is. One type of histone modification which is relevant to this paper is histone acetylation and deacetylation. This involves a chemical group called an acetyl group being added to histones by histone acetyl transferase enzymes, known as HATs, or being removed by histone deacetylase enzymes, known as HDACs. The addition of acetyl groups to histones is associated with the loosening of chromatin structure and therefore gene activation, whereas the removal is associated with the reverse, i.e., the tightening of chromatin structure and thus gene repression. Scientists have already developed effective anti-cancer drugs which prevent the process of histone deacetylation by inhibiting these HDAC enzymes. These drugs are known as histone deacetylase or HDAC inhibitors and are the subject of this publication. In the case of ovarian cancer, poor prognosis has been associated with high expression of a specific HDAC called HDAC2, suggesting that HDAC inhibitors may potentially be an effective therapeutic option. In this paper, the scientists showed that ARID1A mutated ovarian cancers exhibit sensitivity to Pan-HDAC inhibitors, which non-selectively inhibit all HDAC activity. More specifically, they showed the inhibition of HDAC2 activity in ARID1A mutated ovarian cancer cells resulted in increased growth suppression, and they elucidated the mechanism behind this. This mechanism will become clear after we explore the paper, but I will briefly introduce the key players and general mechanism now. ARID1A and EZH2, a subunit of the polycomb repressive complex 2, usually antagonistically regulate the expression of tumour suppressor genes such as PIK3EP1. However, in the absence of ARID1A, HDAC2 interacts with EZH2 and represses its activity, thus suppressing the expression of these tumour suppressor genes. However, HDAC2 inhibition can reactivate the expression of these tumour suppressor genes and their activity in promoting cell death and inhibiting cell growth, leading to enhanced growth suppression in ARID1A mutated ovarian cancer cells. The first experiment that the scientists carried out was a co-immunoprecipitation assay to determine whether HDAC2 and EZH2 interact with each other. The bands on the left side of the highlighted regions indicate that HDAC2 and EZH2 do interact in ARID1A mutated cells, but the disappearance of the bands on the right side shows that if normal ARID1A is restored, this interaction is broken. Therefore, the conclusion from this experiment was that HDAC2 interacts with EZH2 in an ARID1A status-dependent manner. Next, they explored the effect of HDAC2 inhibition on ARID1A mutated ovarian cancer cells. Firstly, they investigated whether ARID1A status affects the response to HDAC2 inhibition by growing the cells with or without ARID1A and HDAC2 inhibition in 3D cultures to observe growth. Here you can see that ARID1A loss enhances the inhibition of growth induced by HDAC2 inhibition. Secondly, they investigated how HDAC2 inhibition affects cell death and cell growth in cells with or without ARID1A by measuring the expression of the apoptosis marker cleaved caspase-3 and the proliferation marker Ki67 respectively. In cells lacking ARID1A, HDAC2 inhibition induces cleaved caspase-3 expression and decreases Ki67 expression, and this effect was not observed in normal cells containing ARID1A. This therefore indicates that apoptosis is induced and growth and proliferation are suppressed by HDAC2 inhibition in ARID1A mutated cells. Following this, the scientists investigated how HDAC2 is involved in the regulation of apoptosis and proliferation. Firstly, they identified genes which are both repressed by HDAC2 and known targets of ARID1A and EZH2, and here they identified the tumour suppressor gene PIK3UP1. They then confirmed via Western blot that HDAC2 regulates PIK3UP1 expression in an ARID1A status-dependent manner, as HDAC2 inhibition only induced PIK3UP1 expression in ARID1A mutated cells, as you can see by the presence of bands on the left, but not on the right. They then investigated what role this PIK3UP1 upregulation could play in the observed sensitivity caused by HDAC inhibition. To do this, they deleted PIK3UP1 in cells with mutated ARID1A with and without HDAC2 inhibition, and measured the expression of the apoptosis markers cleaved caspase-3 and cleaved PARP-P85. They observed a decrease in expression of these markers when PIK3UP1 is absent upon HDAC2 inhibition, which also correlates with reduced growth inhibition observed in 2D and 3D cultures. This suggests that the enhanced growth suppression seen upon HDAC2 inhibition in ARID1A mutated cells is at least partly due to the reactivation of PIK3UP1 and subsequent activation of apoptosis. Finally, the scientists investigated the effects of Pan-HDAC inhibition in mice with ARID1A mutated ovarian cancer using an approved Pan-HDAC inhibitor called SAHA. They showed that SAHA treatment reduced tumour growth and fluid production and improved the overall survival of the mice carrying these ARID1A mutated tumours. To conclude this investigation, the scientists linked the observed in vivo effects in mice with the molecular effects seen in vitro by measuring the expression of Ki67, cleaved caspase-3 and PIK3UP1 in the mice tumours themselves using immunohistochemical staining. Looking at the quantification of these results, there is a significant increase in cleaved caspase-3 and PIK3UP1 expression and decrease in Ki67 expression upon HDAC inhibition, confirming the mechanisms observed in vitro. Overall, this HDAC2 mechanism that the scientists found, which is summarised here, helps to explain how ARID1A mutated ovarian cancer cells are sensitive to HDAC inhibition. Targeting this mechanism using already approved Pan-HDAC inhibitors is a very viable therapeutic strategy, and with the incidence of ARID1A mutations in ovarian cancer, it is urgently required. Moreover, with mutations in both ARID1A and other SWI/SNF complex subunits being prevalent in a large number of human cancers, these results may have therapeutic applications for a wide range of cancer types, an area where future research could explore.